Hello. Today I want to discuss the topic of genetic transfer methods in prokaryotes. I'm Amrita, an assistant professor in Krishna Jayanti University. So as we know that in prokaryotes, we see different mechanisms by which part of a outside genetic makeup or plasmids or genes is transferred uh, from host to the from the from certain cells or even from outside the environment to host cells. So there are different methods of transferring of genetic material from one cell to the other, as well as from the environment to the cell. However, we will focus on the three major methods, which are conjugation, transduction, and transformation. In conjugation, there is generation of a pilus, that is a connection between two cells, and there is transfer of a F plasmid, which is a fertility plasmid, from one cell to the other cell. So where the F plasmid is present, those cells are called as F plus. Where those are absent are called as F minus. F plus has the capability to produce the pilus and transfer the F plasmid to the other cells. And after the transfer, the F minus also becomes F plus. That is how genetic material is transferred from one cell to the other. Now, there are different mechanisms whereby sometimes F plasmid gets integrated into the bacterial chromosome, and then the transfer occurs. As a result, it's not just the F plasmid that is getting transferred to F minus, rather, part of the bacterial chromosome is also transferred, and that is called as HFR, or high frequency recombination. There is also F prime plasmid, where a fragment of bacterial chromosome gets incorporated into the F plasmid. So in the previous case HFR, the F plasmid was integrated into the chromosome. Here in F prime, the part of a bacterial chromosome gets integrated into the F plasmid, and when the transfer occurs, a similar kind of process is observed, whereby F plasmid with part of a bacterial chromosome gets transferred to the F minus cells. Then there is a mechanism of transduction, which is also referred to as a generalized transduction and specialized transduction. In both the cases, there is the involvement of bacteriophage or a virus. So, generalized transduction occurs during the lytic cycle of the bacteriophage. When the phage is assembling new viral particles, it mistakenly encloses part of bacterial DNA, and then when it transfers to the other bacteria, it is carrying that part of the bacterial DNA also to the new host cell. In specialized transduction, it follows the lysogenic cycle, where the temperate bacteriophage integrates its DNA into specific site on the host chromosome. When the phage undergoes activation, it can excise itself incorrectly, taking along only the bacterial genes. So here is the case where, in case of a generalized transduction, this is the bacteriophage, and by mistake, when assembling, the capsid assembly occurs, part of the bacterial DNA also gets inside. This blue color is the bacterial DNA. So mainly it carries its own genetic material, but sometimes the phage DNA by mistake carries the donor cell DNA also, and then when it goes to the other bacteria, it transfers that DNA as well. In the case of specialized transduction, where the lysogenic cycle happens, there the prophage DNA becomes a part of the bacterial DNA. And when the transfer occurs, part of the bacterial DNA has a very high chance, part of the bacterial DNA, to get inside the capsid and enter into the new cell. Then we have the bacterial transformation, where the uptake of DNA takes place from the outside environment, from the surrounding environment. The cells that have the ability to take up the DNA are known as the competent cells. This process was first reported in Streptococcus pneumonia in Griffith. So Griffith was carrying out one experiment, and where he found that certain dead virulent strain was able to give virulence to a live but non-virulent strain, and was able to kill mouse where the injection, where the transfer was happening. It was because the dead virulent strain was able to transfer its viral DNA into the non-virulent alive strain. So that was when it was realized that from the environment also bacterial cells are able to uptake the DNA. Here we can observe that how the gene that is an outside gene, it is often transferred and it gets inserted into the bacterial plasmid using the restriction enzymes. So this is how these are the different mechanisms where how the bacteria can take up DNA from outside and from other cells. So thank you very much.